Hey everyone, my name is Dan Quintana, and over the next few moments I'm going to be sharing with you some of the research, some of the oxytocin research that we've been doing over the past few years at the Norwegian Centre for Mental Disorders Research. Now, when it comes to any new technology, it tends to follow a characteristic pattern when it comes to our expectations of the technology and the performance of the technology. Think about self-driving cars, for instance. When this technology first came out, everyone was very excited, and then these cars start having accidents, and we begin to think, well, maybe these cars aren't as great as self-driving cars, or this technology isn't as great as we thought originally. Now, oxytocin has been following the exact same thing in something that we've characterized within the oxytocin hype cycle. When oxytocin research first came out, everyone was extremely excited. It was based on very solid animal research, and then research comes out, and both researchers and journalists proclaim that this is the miracle molecule. I'm sure you've heard of oxytocin referred to as the cuddle or the hug molecule, but we know now that isn't necessarily the case. So after all these studies, we begin to get these negative studies, and people are now looking at this going, well, this oxytocin doesn't work, and everyone's very disappointed. But now we appear to be entering a so-called period of enlightenment where we're getting a better understanding of what oxytocin can and cannot do. But in order to do that, we still need to do more investigations and take a step back from these studies, or from these clinical studies, to actually look at the mechanisms of oxytocin. A few years ago, Tom Insel came out with a really interesting commentary in biological psychiatry about how can we translate oxytocin from the lab to the clinic. Now, I'm not going to read this out verbatim, but basically there were two things that he highlighted in that we need to firstly understand what is the impact of dose. Currently, most people administer the same dose of oxytocin, 24 IU, and when you ask them, why did you do that dose? They're like, well, that's what the last person did, but that's not really a great justification. And secondly, we don't actually understand and have a good understanding of oxytocin receptor expression in the human brain. Now, these two questions are the two things that I've been investigating over the past few years and what I'm going to be going over today. So the first question is, what is the impact of intranasal dose on cognition, neural activity, and physiology? So in our first study done in neurotypical adults, we had 4 conditions. Our first condition was a low-dose oxytocin paired with a placebo intravenous drip. This was a double dummy design because you can't exactly blind if you're giving people intravenous administration or intranasal. And you'll notice that this spray device looks a little bit different than the typical nasal sprays that you find. This is called a breath-powered device, and we worked very closely with a Norwegian company that's headed up by a nasal surgeon who has designed this nasal spray device. Because the thing is, the typical nasal spray devices that people use were never designed to deliver drugs to the brain. They're only designed to deliver drugs to the nose. This device is specifically designed to do that. So the condition 1 was low-dose oxytocin, 8 international units, and placebo IV. The next condition was a higher dose, 24 international units, which is the traditional dose, and placebo IV. The next one was placebo intranasal administration and a oxytocin IV, which was 1 international unit over slow infusion over 20 minutes. And the final one was placebo. And this was the paradigm that we used. Participants were administered whatever treatment they were given. We waited about 40 minutes, and then we administered the social cognition task, where people were asked, given a series of questions, how angry is this person? How happy is this person, who had a variety of emotions? And then after that, we were collecting MRI during this, and then we also collected resting state and structural MRI afterwards. Now, most importantly, we wanted to look at this comparison between IV oxytocin and intranasal oxytocin. We did some pilot testing, and we discovered that when you actually give 1 international unit of intravenous oxytocin, you can get equivalent peripheral levels compared to intranasal oxytocin. Now, there's a question of how does intranasal oxytocin actually get to the brain? If we have equivalent levels between intravenous and intranasal, then we can actually compare these things. If we get a similar response between intranasal and intravenous, this would suggest that the way that intranasal oxytocin gets to the brain is that it crosses the blood-brain barrier from oxytocin that's circulating in the blood. However, if we find effects only with intranasal but not intravenous, then this would suggest there's a privileged nose-to-brain route that intranasal oxytocin uses. So the first thing was importantly demonstrating similarities or equivalence between intravenous and intranasal oxytocin peripheral blood levels, which is what we did. So for our first result, we demonstrated that only a low dose of intranasal oxytocin was able to modulate the perception of social cues. There was no difference between the higher 24 IU dose and placebo. So we actually demonstrated that the lower dose appears to be more effective when it comes to social cognition. Secondly, we replicated probably one of the most well-characterized results in the oxytocin literature in that oxytocin reduces amygdala activity in response to emotional cues. We found the same thing, but once again, we found it with the lower dose, but not the higher 24 IU dose, and not with intravenous oxytocin, either with the neural response or with the behavioral response. We also looked at pupillometry and the pupillometry response, and once again, we found only effects with the low-dose oxytocin. And what was really interesting was we found similar effects for both social and non-social stimuli. We have this idea that oxytocin is a social neuropeptide, but results like this and results from other labs have demonstrated that it's not necessarily exclusive to social cues. And then we also found that there was a relationship between amygdala activity, or a decrease in amygdala activity, and the pupil response as well. We followed this up with a trial in adults with autism spectrum disorders. And we used a similar paradigm, except we didn't do intravenous and we didn't do fMRI scanning, we just did the social cognition task. The same task, and we found similar results in that only the lower dose of intranasal oxytocin modulated social cognition in this population of adults with autism. So it was nice to see these effects both in neurotypical and psychiatric populations. So this was really, really interesting. But now I want to move on to more recent work that I've been doing, which is looking at oxytocin receptor expression. Now, oxytocin receptors are located throughout the body, but also within the brain. And any intranasal oxytocin that's administered acts upon these receptors in the brain. Oxytocin receptor expression patterns are related to behavior. Research that has been done in prairie voles, which are an excellent model of social interaction, considering that like humans, or like about 3% of mammals, prairie voles are socially monogamous. So they're a great model to use for oxytocin or for social behavior research. The researchers found that experimentally manipulating oxytocin receptor levels in the nucleus accumbens modulates behavior in that it decreases the latency to approach novel conspecifics, but it also increases licking and grooming behaviors. So by experimentally increasing or decreasing oxytocin receptor expression, you can see clear differences in behavior in animal models. What about humans? There has been some research which has taken oxytocin receptor expression in certain sites in the brain, maybe 3, maybe 4 sites, and compared oxytocin receptor expression between these sites. But as we all know, mental states do not reside within a certain area of the brain. These things are networks. So in order to get a better understanding of the relationship between oxytocin receptor expression, we need to look at the entire brain. And that's what we did in our recent study, which was published a few months ago, and which I'm going to go over now. So in this particular study, we used data from the Allen brain dataset, and this is a fantastically rich dataset which has taken samples from up to 300 to up to 900 sites within the brain, and they've done microarray analysis, and what that does is it means we can actually look at the expression of over 20,000 protein coding genes within the brain. And we took this and we created voxel-by-voxel expression maps of over 20,000 protein coding genes. Of course, we were primarily interested in the oxytocin signaling system, but we looked at every single gene and created these voxel-by-voxel maps. The first thing that we found was that the oxytocin receptor gene was highly expressed in the subcortical regions, which was kind of to be expected. But what was really interesting was that we found increased expression in the olfactory region. And the reason this is really interesting is that intranasally administered oxytocin goes from the nose to the brain by the olfactory nerve, which heavily innervates the upper posterior regions of the nasal cavity. This is the first area of the brain where intranasal oxytocin actually enters. And we've actually demonstrated here that it has some of the highest expression of oxytocin receptors. So this is one sample, but we wanted to actually look at, do we actually get similar results from a similar sample? So we looked at the GTEx dataset. This isn't as spatially rich as the Allen dataset. However, there's a lot more participants, but it still allows us to look and to compare oxytocin receptor patterns. And we found that the oxytocin receptor pattern was highly related between the two data set. So it was a nice validation that it wasn't necessarily a fluke, that we found it in this data set, and we demonstrated it in a second data set. Now, because we did microarray analysis, or analysed a microarray analysis, we were able to correlate oxytocin receptor expression along with other key genes which are key for oxytocin signaling, like CD38, which regulates the secretion of oxytocin, with other important genes. And we specifically singled out dopaminergic and muscarinic acetylcholine genes. And we found that these oxytocin genes were highly expressed. The correlation between the expression patterns was extremely high between these gene sets. Okay, so what do we find? So we've created these oxytocin receptor maps, created it for the oxytocin receptor, for CD38, which regulates the excretion of oxytocin, and also the structural gene for oxytocin, which is just called the oxytocin gene. But then, how are these things related to mental states? So to do this, we use the Neuroscience tool, and this is an incredible tool which I think has been mentioned earlier today. And basically, you take a specific mental state, let's say pain, and then using a corpus of over 14,000 fMRI studies, you can automatically extract coordinates and create a summary activation map of a given mental state. And with this tool, you can either use forward inference, so you can take a mental state like pain and then generate the summary map, or you can go the other way around and you can actually get a map and see which mental states are most highly correlated with this map. And that's what we did with oxytocin. And this is what we found. These are the most highly correlated mental states that we found with the oxytocin receptor, with CD38 and OXT. And what was really interesting is that you can broadly characterize these mental states as anticipatory, appetitive, and aversive. And these mental states aren't necessarily social. We have this idea that oxytocin is a social peptide, but what's interesting is that a lot of these mental states aren't. And look, one of the most interesting ones that we found was the mental state of taste. And that's really interesting considering the emerging research that suggests that oxytocin actually might be involved with appetite and energy regulation as well. So these were the most highly correlated sites that we found. So, like I mentioned before, oxytocin was originally conceptualized as a pro-social hormone, the cuddle hormone, the hug hormone. But later research actually demonstrated that that is not necessarily the case. And more recently, we've conceptualized oxytocin as a social salience hormone, in that it increases the salience of social cues in our environment. But now, with this research, it's looking even more broad than that it seems to be a more broad regulatory hormone. So the story is changing a bit, but that's good because we're looking at new evidence and refining our theories, which is incredibly important. So we did find these high correlations with the oxytocin receptor expression and with specific mental states. But this might not mean anything, because say we have a high correlation between the oxytocin receptor and the mental state of taste, for instance. What if there are, say, another 5,000 genes which are even more strongly related with the mental state of taste? Well, in order to look at this, we actually looked at these distributions and ranked how highly the oxytocin receptor expression was compared to all other 20,000 protein-coding genes. And what we found was that with almost all of these mental states, the oxytocin receptor expression was ranked within the top 1% to 2%. So not only was there a high significant correlation between these, but when you look at these specific mental states, the oxytocin receptor was among the most highly ranked compared to over 20,000 protein-coding genes. Okay, well, we have the expression patterns, voxel-by-voxel expression patterns of over 20,000 genes, so we may as well look at which genes are most highly correlated with these patterns. And this is what we found, that the most highly correlated genes seem to be linked with metabolic regulation, which is really, really interesting, I think. And the most negatively correlated genes were actually linked with two genes which have been characterized to be involved in schizophrenia. So what are our take-home messages? Well, the first one is that a lower intranasal dose may be more effective. Now, there are two possible reasons why we found this. Firstly, perhaps a higher dose cross-reacted with vasopressin, which sometimes has sort of a yin and yang type conceptualization that it does the opposite to oxytocin. The other possible reason for this is that our optimized nasal spray that we used almost worked too well, and an 8 IU dose using this device actually worked much like a 24 IU dose from a traditional device. So those are two ideas that can potentially explain our results. The second take-home message is that intranasally administered oxytocin reaches the brain via nose-to-brain pathways. We demonstrated that although we had equivalent effects between intravenously and intranasally administered oxytocin, we only found behavioral, neural, and physiological effects with intranasal oxytocin, not with intravenous oxytocin. So it doesn't seem to cross the blood-brain barrier, it seems to enter the brain via the olfactory nerve fibers. And finally, that the oxytocin signaling system modulates cognitive state processes that are involved in complex human states, which are related and which work in concert with other signaling systems as well. So we're going to have questions at the end, obviously, but if you do have any other questions, please send me an email, and I've posted the links to the slides and to the references on an OSF page here. And finally, I'd like to give a big thanks to the team and to my funder, the Novo Nordisk Foundation. Thank you for your attention.